Hi, I would like to thank Biotechnologierådet for organizing this conference and for inviting me to contribute to the discussions here. My name is Marelle Kaufmann and I am a criminologist and a cultural scientist. My work looks at how digital data and technologies, but also biodata, come to matter in security practices. Hence also this nice material depiction of the DNA double helix. But I also look at how technologies give rise to critical engagement with security practices in return. Many of the processes I study, whether that's emergency management, policing, hacking, the use of secret codes, lab work, or art projects, help me realize that digital data and technologies change the ways in which we create knowledge, and they're changing how we relate to each other. We do hear this a lot, especially at this conference, but we cannot say it often enough. Digital data analysis holds quite a powerful position in social and scientific settings. But how and why digital data analysis reach that position is not always easy to see. So with my newest projects, I want to understand the role of two concepts in data analysis better. And these concepts are central to this conference: life and evidence. Let's first look at the concept of life in relation to data analysis. When I try to understand the role of life in data analysis, I do not mean that data or analytic tools have consciousness. This is no attempt to associate digital data analysis with AI, where humans would go beyond biology. Quite the contrary. I develop an argument for the importance of bios, specifically human life, in data analysis. And in order to study how digital data and life influence each other, I have gotten more and more into the analysis of life cycles. We can study the life cycles of humans and other organic life forms, but also of tools, data, and the relationships between them. And when we observe how life cycles overlap and intersect with each other, they make us aware of the processes of coming to life, being in formation, of being discarded and composted, of acting back and of coming back to life. Most important, if we manage to trace how the life cycles of humans, digital data, and tools influence each other, we can identify how we want to shape our lives with data analysis. Quite an important question of this conference. The first thing we can observe is that the life cycles of humans and data analyses increasingly meet. Not only are any data generated from humans, but digital data also shape human life in return. The data life cycle is here a helpful tool because it follows data through its different stages, from creating data to processing, analyzing, preserving, accessing, and reusing it. In the best case, the data lifecycle inspires reflections about the making of data. Who actually generates data? Under which circumstances and how? Part of the data lifecycle is also how researchers read and interpret data, how researchers assign meaning to data. When we try to identify the ideas that guide cleaning and processing data sets, for example, we can trace the politics of data analysis. And understanding these ideas helps us discussing the consequences data analyses can have for human lives. I have studied these processes, for example, in predictive policing, where data cleaning processes have very concrete effects on the ways in which cities are being patrolled. So when we follow data through their life cycles, we also understand how data themselves become lively. What does that mean? Understanding data as lively actually contradicts the original meaning of data as something given. Rather, when we follow the life cycles of data sets, it's almost appropriate to say that data are not a given, but in a state of becoming. There is a growing understanding that data become lively. Deborah Lupton, for example, shows us how data circulate across different systems, how they can be reused for different purposes. And when they're used across different domains or are translated into different contexts, data live their own life because they become quite independent of their original owners. Other scholars have focused on a different aspect of lively data. They have shown that data sets can actually act back when they, for example, create surprising effects. I found, for example, that some predictive policing teams have correlated astronomical data with crime data and found that their algorithms started predicting crime patterns according to the phases of the moon. No tested explanation exists for this correlation yet, but it goes to show that data sometimes can start to act back or act strange or surprise us or become lively. A third party in these lively interactions are the methods and tools used to analyze data. Digital methods and tools are transforming research to such an extent that Mike Savage reminds us that analytic tools too live lives. The life cycle can once again help us to understand the ideas embedded in digital methods. The life cycle enables us to examine, for example, the databases and parameters an algorithm is trained on before it is tested and used. So the life cycle also makes us ask whether and how methods and tools die, how they're reused, or if parts of them are recycled. So taken together, life cycles help us seeing the intersections with the lives of other tools, of data, and humans. And this is very central if we want to understand how DNA comes to matter in police work. If we can see that digital data, analytic tools, and human lives influence each other, the next step is to ask what this influence does to the idea of evidence, specifically DNA evidence. The relationship between digital data analysis, DNA, and evidence is changing fast, but there are quite a few things we already know. For example, DNA itself may not be as scientifically evident as it's often presented. Since the 1990s, we know that there are science wars about methods in DNA sequencing. In particular, studies about the identification of an individual from a DNA sample and the use of expert knowledge have called DNA status as the gold standard of forensic evidence into question. Many studies signal that the credibility of methods and the evidence they produce is also a socio-political accomplishment. And still, DNA's association with science and evidence is pretty persistent. In addition, DNA analyses have become more and more integrated with digital technologies. We see one of those here on the slide. They further emphasize the relevance that DNA has in the forensic system today. Scholars have considered important social, legal, and ethical aspects of such innovations in digital DNA technologies. Matthias Wienroth is a specialist here, and Ahmad Msharek's work on phenotyping is an excellent study of the ways in which DNA phenotyping targets populations instead of individuals, and it even produces new legal notions of race. So in extension of these works, I want to ask questions about the ways in which digital technologies shape DNA evidence. The need for this research is also voiced by political, legal, and societal actors. Norway's federal prosecutor general declared that there's a need for in-depth research about DNA processing and other digital technologies in legal genetics. The UK Science and Technology Select Committee expressed serious concerns about the scientific validity of some forensic science fields and called for the development of a better understanding of digital forensics. So when I try to address these gaps, I want to remind us how important it is to understand the life cycles of digital data analysis in the making of DNA evidence. I'm only starting these projects, so I cannot yet provide you with empirically based insights, but I will give you an overview of the developments that take place in digital forensics today and formulate what is at stake. In this talk, I will focus on three fields that need our attention. First one, how does smaller, more mobile and specialized hardware influence the production of DNA evidence? Why is this a relevant question to ask? Because we have smaller and more mobile computing technologies today, we see a change in forensic procedures. Biological material and investigators no longer move to the laboratory, but the lab goes into the crime scene. This also involves more collaboration between public and private service providers, because each step in the analytic procedure is more and more professionalized. The collaboration in DNA analysis is very complex. So it is important to understand how the fact that hardware can move into the field changes the production of evidence. That's really crucial, because mobile hardware is the first step in translating organic material into digital information. And this process starts with machines that extract and quality control DNA. We see here the Agilent 2100 bioanalyzer, but also Fluidigm C1 on the slides. And this process continues with technologies that interpret and visualize this information, which can be, as these technologies can be as mobile as an ordinary computer, as we see on this other picture here. Together, these technologies aim to give indications of what a potential victim or perpetrator could look like. And this means that forensic results may be produced faster, but it also pressures forensic biologists into providing results based on the preliminary visualizations of these tools. And as we have heard, one of the consequences is that forensic analysis then moves from the identification of individuals to rendering a whole group suspect. The forensic community is aware that such preliminary outputs may raise doubts in the production of evidence, since they only indicate tendencies. Information at this stage is speculative. However, visualizing digital information can also stabilize the direction in which DNA evidence is being interpreted. So through phenotyping, phenotyping is not yet used in Norway, but we need to document and discuss the influence of new hardware on the forensic process and provide a new understanding of DNA as evidence. The second question is, how does a change in the collection, availability, and algorithmic analysis of DNA influence the production of DNA evidence? So why is that question important? Biological information, including DNA, does not exist per se. Such data are taken from people or items. They are collected and stored in various databases. They are accessed and analyzed by various parties and for different purposes. In the past years, we have seen the rise of DNA databases, like, for example, 23andMe. There's also HapMap. We also see here the 1000 Genomes Project. This not only leads to a general increase in available DNA data, but also changes the access that the police, scientists, and lay people have to such data, and creates very interdisciplinary settings, which we still need to understand. So due to this rise of DNA databases, the data available tend to become more comprehensive and diverse. And here we already see similarities to trends in big data analyses. Data sets are not subject to function creep, as we like to say, but their function is the creep. So the supposed advantage of big data sets, even with DNA, is that the same data can be studied within many analytical projects. So no data is irrelevant. In the same way, there is no such thing as junk DNA for those who analyze biological material, since even the area around or outside the coding regions of the genes can tell its analyzers something about a person's origin. At the same time, there is a legal debate as to whether biological material is personal information and whether its collection and analysis should be more tightly regulated. In many countries, including Norway, DNA becomes personal information only when it is analyzed and related to an identified person. However, various legal entities are currently evaluating the status of biological material as personal information. In general, one can say that the growth of DNA databases broadens biosurveillance. The specificities of databases therefore need to be better understood. What information actually is collected and stored? How is it stored? As well as who will be allowed to work with it? The availability of DNA in digital databases also leads to the increasing use of algorithms within these data. That's the second part of the question. So that's why we're asking how information that is available in biobanks is actually analyzed. DNA data are processed by software, and that is algorithms which search information for patterns. And these algorithms are programmed in specific ways, which determines how they identify results. For example, software designers need to decide on the parameters the algorithm uses to translate DNA samples into graphs or other visualizations. The software needs to assign visual values to DNA samples, despite uncertainty about what can be considered noise and what is actual signal in the sample. The result of such algorithms are thus like a real production of evidence, in the sense that some patterns would maybe not be found with a different algorithm. So it is crucial to get a better understanding of what happens when DNA information is analyzed by algorithms. The increasing sophistication of algorithmic and of algorithms could produce future evidence too, with their analytic power being used to find out who may become criminal, and that touches on the question of profiling again. And the third and last question, how does the ability to alter human DNA influence the production of forensic evidence? This question focuses on what is probably the most revolutionary shift: changes in information per se. It is not just people, instruments, and processes that influence the production of DNA evidence. The very object that indicates evidence has begun to object, as Latour likes to say it, within the production of evidence. Remember the life cycle of data and how data becomes lively. So we need to better understand how the ability to change DNA also changes its status as evident information. In the last decade, genetic engineering, the manipulation of genes via biotechnology, has grown, for example, with the CRISPR-Cas method. This means that DNA, the object that is considered as evident per se, can be changed by means of digital technologies. Working with DNA has become relatively cheap and easy, using online programs, for example. Currently, this includes using biobricks or building bio-objects. At the same time, one has to take into account that DNA has a big storage capacity, and the way it works with the body and the body's environment is so complex that people often use the language of orchestration to explain it. So this means that making something as complicated as DNA is not really going to happen anytime soon. But changing DNA requires professional training and expensive laboratory equipment. Nonetheless, the ability to change the very object that seemed evident or given may also raise new insecurities within forensic processes. Even if changed DNA is unlikely to be planted at crime sites, the trend to contest the reliability of forensic science's most significant player in establishing evidence is worth investigating. So having said that, a careful analysis of these trends also needs to take account of the challenges that exist already today. So for example, when DNA is mixed up, contaminated, or not analyzed promptly, when results are vague or inconclusive, or when it cannot be analyzed because its carrier is difficult to store. So nonetheless, we see that the ability to alter or change human DNA is a powerful indicator of the role of matter, of the different lives and agencies at work when something is established as given or as evident. And that is why what I think is worth investigating when we want to speak about DNA and police work. And I thank you so much for listening.